This video will be demonstrating the extended flow cell clean on a fax area. This procedure is used by BD field service engineers to clean the flow cell when a normal flow cell clean with BD detergent is not working. They will use this when you have wide CVs or a lot of noise from improper cleaning after using the cytometer. To get started, we will open the door to the sort chamber. Once open, we will remove the two high voltage plates. To remove the plates, we will use the plate removing tool. You will hook the tool around the plates and pull. We will remove both plates. We will remove the locking lever and then finally the nozzle. While you're doing the extended flow cell clean, it's always a good idea to sonicate the nozzle. We are going to put the nozzle on a 5mm fax tube and submerge it in DI water. We will take it over to the sonicator where we will place it into the sonicator and then put the lid on. We will activate the sonicator and start the sonicating process. Sonication will clean out the nozzle's orifice, so if there is a clog or some buildup, that will be removed. This will normally be done during the morning setup, but this is something extra that you can do while you are cleaning. We will be running a solution of 50% Contrad 70 for the extended flow cell clean. This is a bottle of Contrad 70. You can use a transfer pipette. This is a 5mm fax tube, so there is 2.5 ml of Contrad and 2.5 ml of distilled water. We're going to place the 50% Contrad 70 solution in the tube holder in the bulk injection chamber. I'm going to place a paper towel around the aspirator drawer because this can potentially get messy once we start the stream. In Diva software, under cytometer, we are going to select cleaning modes and then clean flow cell. Select OK because we have the cleaning solution already in the bulk injection chamber. When the clean flow cell is run, it will put 50% Contrad all throughout the sample line and the flow cell. We will run multiple clean flow cells until the tube is gone. As you can see, we have gone through practically all the Contrad in the tube. Next, we will let the Contrad sit in the flow cell for 5 to 10 minutes and soak. After the 5 to 10 minute soak, we are going to load an empty tube into the bulk injection chamber. The idea behind this is we are going to push air up through the sample line and into the flow cell. This will agitate the Contrad in the flow cell, creating bubbles and scrubbing out the inside of the flow cell. When we run the clean flow cell, we will see bubbles in the flow cell and the Contrad being agitated. Once the tube unloads, we will start the stream. There should still be nothing in the sort block, no nozzle, locking lever, or plates. This is where it can get messy, so have a paper towel around the aspirator drawer to catch any residual spray. Once the stream starts, we will load the empty tube again. This will help to push out the residual Contrad in the line and flow cell. You will only need to run the empty tube for a couple seconds. Unload the tube and turn off the stream. Now you will need to drive all the parts of the sort block: the nozzle area, the locking lever area, and the sort block itself. When I'm drying out the areas of the sort block, I will take a cotton swab and dry the nozzle and locking lever areas. I will also dry out the underneath area of the flow cell. I will make sure that in Diva software there are no visible drops in the stream camera, which can affect sweet spot and your stream view. Then I will dry the rest of the sort block out and start to reassemble the nozzle, locking lever, and plates. The nozzle has been removed from the sonicator and dried. We will start reassembling by pushing the plates back into place. The plates just push into position. Next, we will install the locking lever. And finally, inserting the nozzle and locking it in place. With everything back in place in the sort block, we are now going to turn on the stream. With the stream on, now we can run CST performance check again to see if there's an improvement from the cleaning. This concludes our video for the extended flow cell clean on a BD Fax Area 2. Thank you.